All right, so here is our little update. A couple of interesting papers. One of them is, is arriving just in time. So this was a paper from that same group. So we've got Mike Diamond and Sean Whelan on there, and these are people that are associated with the virus. It's VSV, vesicular stomatitis virus, the one that's shaped like a bullet bill from Super Mario. And they have, instead of the normal protein that's on the outside of that, they've taken that off and they put on the SARS coronavirus spike. So now it gets into cells that SARS can get into, but it doesn't cause SARS disease because the virus inside is a really weak horse virus that does not do well in humans. But they can use this to investigate exactly what would happen. And this is also a pretty decent vaccine candidate that I would like to see tried out. Yeah, may not work, but at least this one's got a better chance, I think. Anyway, this paper is about how the virus gets in. And so what they're looking at is intestinal cells. So this is something we've been talking about. We know that there's evidence of virus in the virus coming out in feces. There hasn't been, at least as far as I've seen, any direct evidence of infected cells in the intestine. And so people have been suggesting that this is all virus that's coming from some mysterious other organ that I'm not even sure if it exists in anatomy books. The mysterious space organ. That the virus is coming from somewhere and just being flushed out down the enteric tract. That's the idea. I always think that's probably not what's happening, because you wouldn't get this amount of virus there and it wouldn't be that consistent, is what is the logic. But again, you don't want to answer these things by logic, you want to answer them with science, right? So let's answer it, or rather they have answered it. And they have tested it out and found that there are a lot of cells. They're called enterocytes. They're the cells that line the intestinal tract. And a lot of these have very high amounts of ACE2, the main receptor, which we already knew. And those are the ones that get infected. And because you're good students of science / this channel, you know that it takes more than just ACE2. It also takes one of those proteases. And so they found that, yes, indeed, there is TMPRSS2 there, and that can work. But if you take away TMPRSS2, there's another one called TMPRSS4, which is another protein-cutting enzyme that is also going to be on the cell surface in these same cells. And yeah, there's a lot of TMPRSS4 in the gut. It's particularly highly expressed there. You also find a fair amount of it in the liver and a couple other organs in the body. So it's pretty widespread. I think it's in the kidneys as well. These are organs that you probably could infect very much with SARS if you ever had SARS in the bloodstream, which thankfully seems to be something that does not happen often and probably only happens in sort of late-stage fatal cases. But anyway, they show that, yeah, TMPRSS2 and TMPRSS4 are now totally acceptable ways to get this virus into intestinal cells. And they did it with their cool little bullet-shaped pseudo-coronavirus. And so, yeah, that's a neat study. It's out there. It came out in, I want to say, Science. No, Science Immunology. Okay, it's one of the lesser science journals, but still pretty darn good. So there you go. Yeah. And this is pretty good evidence that SARS coronavirus at least can infect the intestine directly. And so what I think you're going to see is people will gradually assimilate this information, and we can put to bed the idea that the virus is coming from somewhere and just ending up there. It's probably going to be a direct intestinal infection, and frankly, how would you stop that from happening given the amount of virus that you're likely to see in the intestines? Yeah, or the amount of receptor, yeah, in those cells. Yeah, it's just it's something that can and probably will happen, and now we know does, at least in the laboratory. So it's not confirmation from inside of a patient, but I don't need to see them cut up any dead people at this point. I think we can reasonably work with this hypothesis. So yeah, there we go. Thanks very much. This has been Ask Dr. Ben.